Okay, let's finish up chapter 12. Here we go. So we talked about what cardiac output is, heart rate times stroke volume. So let's talk about the influences on cardiac output. Basically, cardiac output, the amount of blood pumped out of the heart every minute of time, generates a pressure, and that generates a pressure in the systemic circuit and pulmonary circuits in the entire cardiovascular system. So the heart is responsible for creating a pressure gradient that blood is able to flow down. So blood pressure is basically created as blood exerts a force against blood vessels, and that pressure changes as the ventricles pump blood out of the heart or fill with blood as blood makes its way back to the heart. So arterial pressure is much higher on the arterial side, so think arteries, than the venous pressure. And that basically, blood pressure is the pressure in the entire cardiovascular system that is influenced by what's called peripheral resistance. Think resistance to blood flow in blood vessels. If you have a really large tube, it's pretty easy for fluid to flow through it. But if you try to push that same amount of fluid through a smaller tube, it gets a lot harder. The pressure through that tube that allows fluid to flow through is much higher. So basically, resistance to blood flow increases as the vessel size gets smaller. So think about vasoconstriction. As the blood vessel constricts and the passageway through that blood vessel, the lumen, gets smaller, it's harder to push blood through there, and the pressure that blood exerts against the wall of that vessel is even higher. But capillary pressure is pretty low compared to arterial pressure and venous pressure, and that allows time for the capillaries to exchange nutrients and wastes and gases. And what can affect that is basically the neural and hormonal regulation. So think sympathetic, parasympathetic nerves, but also epinephrine and thyroid hormones that we just talked about in the first section of this chapter affect heart rate and stroke volume, but can also affect peripheral resistance. And then venous pressure affects a characteristic called venous return, the amount of blood that is being sent back to the heart to the right, basically arriving at the right ventricle every minute. So how much blood is being pumped back through the superior and inferior vena cava back to the heart? That's venous return. How much blood is flowing back to the heart from the veins, or how much are the veins returning to the heart? How much blood? And venous return affects cardiac output because basically venous return, how much blood is being pumped back to the heart, influences how much blood can be pumped out of the heart with the next contraction. So if you fill the heart, basically venous return affects how much blood fills the heart, and then the ventricles can contract and pump that blood out. So the more blood you send back to the heart, the higher the venous return, the greater the stroke volume in the next contraction. Blood pressure is really important because blood flow is proportional to blood pressure. Basically, how much blood is flowing per minute of time through blood vessels. And it's inversely proportional to resistance. Remember, as resistance increases, likely the size of the vessel decreases, increasing resistance and decreasing blood flow. And blood flows down a pressure gradient. Basically, from one end of the vessel to another, where there's high pressure versus low pressure, blood will flow down from an area of high pressure to low. And basically, the base of the aorta pressure is highest, and at the distal end of where you have capillary beds, you have very low pressure. So blood will flow down that pressure gradient. And during ventricular systole, contraction, pressure increases. That's called systolic pressure. It's called systolic pressure because that pressure is the highest pressure in the experience in blood vessels because the heart is in systole. And then ventricular diastole pressure falls. So you have a blood pressure that you're able to measure. Do you know your blood pressure? My resting blood pressure is like 90/60 beats millimeters of mercury. A normal typical blood pressure is about 120/80. There's an example here of 120/90, and I'm going to place in here millimeters of mercury because that is the actual unit, and I want to be specific. The difference between systolic pressure and diastolic pressure is called the pulse pressure. Basically, as a result of the heart pulsing, contracting, Pressure increases from diastolic when the heart's relaxing to systolic when the heart's contracting. So take a look at this. Right here is your systolic blood pressure, right? The heart contracts, the ventricles contract, and pressure increases. The ventricles relax and pressure decreases, and that happens over and over and over and over again. But as vessels from larger arteries get smaller and smaller and smaller and reach the arterioles, basically, pressure drops drastically because the vessels are branching from the aorta. Every vessel in the body branches off of the aorta in some way. Then those vessels that branch off the aorta continue to branch and branch and branch and become smaller and smaller and smaller and distribute blood through tons of vessels instead of just a few. So that drops the pressure dramatically. And then once you hit the capillary side, the capillaries between the arteries and the veins, pressure is relatively low, but even lower on the venous side, so that blood is continued to drive through the capillary bed and into the venous side of circulation. And then that one measurement that we talked about, pulse pressure, is the difference between diastolic and systolic blood pressures. Within the capillary bed, there's a couple of mechanisms that occur: filtration, diffusion, and osmosis. Remember, you looked at osmosis a lot in lab and did a whole physiology lab activity related to osmosis. But basically, the capillaries have a hydrostatic pressure, a fluid pressure, that forces substances out of the capillaries and into the tissue. So that blood pressure within the capillaries is the capillary hydrostatic pressure, and that's the driving force that pushes water and solutes out of the bloodstream and into the interstitium. So this is the capillary side, this is the tissue side. So this pressure is relatively high in the capillaries and forces solutes through the epithelial tissue and into the interstitium. So you can drive basically gases, oxygen out, nutrients and water can be driven out down a pressure gradient into the interstitial spaces between the endothelial cells. There's little gaps in the endothelial cells that can force small solutes out, and water. These are little water molecules. So what also can occur is filtration. Basically, the movement of water and solutes out of the capillary predominates near the arterial end of the capillary, and then as pressure falls from the arterial side to the venous side, reabsorption can occur. And reabsorption is the movement of substances from the tissue back into the blood. And that's basically by way of osmosis. Water can move, and solutes can move back into the capillaries as well. So much more water is filtered out of the blood and sent into the tissue than is reabsorbed. So that water that gets filtered out of the blood and into the tissue returns back to the venous circulation by way of lymphatic vessels. Remember, we talked in the lymphatic system in the previous chapter about fluid movement out of the blood and into the tissue, and then from the tissue, that fluid doesn't move necessarily back into the blood. It actually moves into the lymphatic vessels, and lymphatic vessels carry that lymph fluid, back to eventually the subclavian veins, right, by way of the thoracic duct and the right lymphatic duct. So here's basically what gets filtered out: 24 liters a day. A lot gets reabsorbed, but the difference here, 24 minus 20.4, gets sent into the lymphatic vessels, and back into the venous circulation. We talked about this cardioacceleratory area previously, these neural mechanisms that can cause short-term changes. The cardiovascular center is located in what area of the brain? Do you remember from the last part 2 of this chapter? Located in the medulla oblongata. There's the cardioacceleratory area, the cardioinhibitory area, that increase and decrease heart rate, respectively. But there's also a vasomotor center that controls vasoconstriction in blood vessels. Remember, if you vasoconstrict a blood vessel, you decrease the size of the passageway for blood to flow through that vessel, and that increases resistance to flow. Blood pressure increases, and flow decreases. And you can vasoconstrict or vasodilate to certain tissues to inhibit blood flow or increase blood flow where needed. If you have ever stood up and got lightheaded, You stood up too fast. So maybe you jumped out of bed in the morning and you got super lightheaded and had to stand there for a second or two and go, whoa, until you got your bearing about you. Well, there's baroreceptors that we talked about once upon a time. And there's locations of baroreceptors in a bunch of different places in the body. But one particular area was in the aorta and the carotid arteries. So there's baroreceptors there that they are a specific type of mechanoreceptor that responds to stretch in a cell membrane. So these baroreceptors provide our body with a reflex to maintain blood pressure within a normal window. So if blood pressure increases too much or decreases too much quickly, the baroreceptors respond and send a signal to the medulla oblongata, where those cardiovascular centers are located, to bring blood pressure back down or back up, depending on the original stimulus. So the baroreceptor reflex is triggered in response to changes in blood pressure. And those changes in blood pressure that stimulate the baroreceptors, these sensory receptors that are monitoring blood pressure, they send signals to the vasomotor center, the cardioacceleratory and cardioinhibitory areas in the medulla oblongata. Those are integrating centers. The medulla is the integrating center, the location of these integrating centers for blood changes in blood pressure. And those centers will then send motor information back to the heart and back to blood vessels to respond to changes in blood pressure. So here's what happens. Let's start here. Decrease in cardiac output occurs and maybe vasodilation. So blood pressure drops. So if cardiac output decreases, less blood is being pumped through blood vessels, so the pressure will drop. And vasodilation increases the size of blood vessels, also decreasing pressure. So if blood pressure drops, that's disturbing homeostasis, right? Let's do it actually down here. So blood pressure falls. The baroreceptors are inhibited. The vasomotor center basically receives that signal from a drop in blood pressure. The baroreceptors stop firing action potentials to these centers. And those centers then say, okay, what are we going to do about that? Blood pressure has dropped. We need to bring it back up. To do that, blood vessels from the sympathetic nervous system stimulate vasoconstriction. So the vasoconstriction occurs, decreasing the size of blood vessels and increasing pressure, increasing resistance. And cardiac output increases by increasing heart rate and the force of contraction. So when blood pressure drops, heart rate and cardiac output go up, vasoconstriction increases, and that brings blood pressure back up to normal. Well, the opposite happens. So here's where we start. I started wrong over here. Sorry, guys. When blood pressure rises, the baroreceptors fire action potentials and communicate to the medulla, where these cardiac centers are located, hey, blood pressure has increased. We need to bring it back down. To do that, motor nerves head back out to the heart and to blood vessels to stimulate vasodilation and a decrease in cardiac output, both of which bring blood pressure back down. So if you stand up and get really lightheaded, cardiac output decreases and blood pressure drops. Basically, when you stand up really quickly, blood pools into your lower extremities. Gravity pulls it down. And that stimulus is received by the baroreceptors as a drop in blood pressure. They send a signal to the cardiac centers in the medulla to increase heart rate and increase vasoconstriction to bring blood pressure back up. And it happens in just a couple of seconds. When you stand up and get lightheaded from standing up too quickly, it only takes you maybe 2 to 5 seconds for that to come back to normal. That's how fast your baroreceptors are working. In the short term, the baroreceptor response is a very, very quick response to changes in blood pressure. But also you have hormonal controls that can help with that within a couple of minutes. So a couple seconds, the baroreceptors respond. A couple of minutes, the adrenal medulla can release epinephrine and norepinephrine, and that can increase cardiac output and also constrict certain blood vessels, which increases blood pressure. But in the long term, hours to days to weeks and months, the kidney's involved in blood pressure regulation. There's a number of different ways that the kidney can be involved in the regulation of blood pressure. Antidiuretic hormone is actually produced from the hypothalamus, secreted from the posterior pituitary lobe. and can act on the kidney in the collecting duct, a structure we'll talk about later. There's other enzymes. Erythropoietin increases red blood cell production. That can increase the viscosity of blood. So if an increase in blood, basically the kidney is your long-term blood pressure regulation organ. Very quick response is baroreceptors, so within seconds. A couple of minutes, the adrenal medulla can help that along, increase blood pressure. But over hours and days, long-term regulation involves the kidney. There's a summary figure of that and how both short-term and long-term mechanisms can work. In response to high blood pressure, an increase in blood volume or pressure stretches the walls of the heart. And there's another hormone called atrial from the atria, natriuretic, this has to do with sodium, peptide, that can reduce blood volume and pressure. So there's some responses that can lower blood pressure or increase blood pressure. This is if blood pressure is too high, how to bring it back down. And there's another summary slide related to that. Again, natriuretic peptide has to do with sodium loss. Cardiac arrhythmias can occur, and I'm going to point out a couple of them, but not all of them, which is basically an abnormal pattern in electrical activity in the heart. You can get, so we have the pacemaker of the heart in the right atrium, but that pacemaker, remember I said all cells in the heart are autorhythmic. So they can cause some problems. In a healthy person, you can see what's called premature atrial contractions, where the atria contract before the SA node actually fires an action potential. So right here's normal, P wave, QRS complex, T wave. But look here, P, QRS, T, and then all of a sudden an extra P wave, or the SA node fires a little bit quicker. That can happen in normal, healthy people in response to stress or caffeine. Certain drugs can change the permeability of the pacemaker cell membranes and tell them to fire a little bit faster accidentally. So that can change the rhythm. You can just see a firing of that a little too quickly. If the SA node breaks down completely, you get what's called atrial fibrillation. So look at this. You have this kind of quivering here of the electrical signal and then QRS complexes. So the AV node is working properly. And it can continue to work properly if the AV node or if the SA node shuts down, but that's not. It makes for a much less efficient heart. So basically, what happens is the atria just quiver. There's no SA node to say contract now, contract now, contract now at a regular interval. So all the cells of the atria just try to contract on their own with no coordination. Those arrhythmias in the atria aren't nearly as severe as in the ventricle. A premature ventricular contraction, single PVCs are common, but with an increase in frequency, that can cause problems. So you can get a premature ventricular contraction occasionally. It's shown here where you see P, QRS, and right after the T wave, there's the QRS complex again, no P wave. So the ventricles are contracting a little too quickly, or there may be something like an ectopic pacemaker. An ectopic pacemaker is a set of cells that fire action potentials outside of the SA node and stimulate the ventricles to contract prematurely. Ventricular fibrillation can be fatal. It's basically cardiac arrest. Without fixing it, without basically shocking the heart back into a normal rhythm, this is what it looks like. No P waves, no QRS complexes, nothing. The ventricles are in big trouble. There's nothing there to coordinate contraction relaxation. And if there's nothing there to coordinate contraction relaxation, then the heart won't function. The ventricles don't function. And if they don't function, then you could die. Okay, that is it for chapter 12. I think we are done with this unit and you have an exam coming up, so good luck.